go back over a second time, maybe more, how shall I put it, the kinetic schemes we were talking about yesterday, because at the end, or not at the end, somewhere in the middle, I want to show you what happens when you do a simulation of this kind of kinetic proofreading. And you can play with it yourself if you like. So I'd like to go through the algebra in a simplified way so that you're very clear on what's going on here. So we're algebra for sorting schemes. So just to review, the simplest sorting scheme is that we have the selection enzyme. It works on a substrate. The substrate binds. And then either it goes back, it says never mind, doesn't do anything, or it makes the product. You have some polymer chain that's being created, or you're charging up a transfer RNA or whatever. But anyway, there are two ways. Once the enzyme has glommed onto the substrate, it either gives up or goes ahead and makes the product. And we have some rate constants for all of this. Okay, and we can do this. For simplicity, we're saying there are two options, right? They're equally present in the cytosol, and one is the good option, one is the bad option. What I really want to do is to select this guy, and in the end, what really makes the selection possible is that at this stage, there is this off rate. He basically says, okay, no, I don't want to do this, and there's some pretty much common reaction rate for making the product. And if you have stuck the wrong thing on your enzyme, making the product is a bad idea. So the option for correcting comes from basically not making the product until it has had time to think about it and maybe kick off the wrong one. So anyway, this off rate is going to depend on whether it's the good substrate or the bad substrate. And ideally, or rather what we'll see is that most of the effect comes from the off rate for the bad thing is big compared to the off rate for the good thing. Seems reasonable. Okay, and so the on rate of making the enzyme substrate complex is something we're not going to worry too much about its dependence on what is the specific substrate. Of course, it will depend on its concentration. We're going to imagine that since this is basically it has to do with diffusion, the things that are diffusing onto the enzyme, they diffuse at pretty much the same rate, whether it's the good guy or the bad guy. That's not doing any selection for you. Okay, so you set this up. You're in solution. You've got a concentration of A and concentration of B, presumably are about the same. And what happens? Well, there are these reaction equations. And we ask, what happens if you go to steady state? All right, just let it run. Well, okay, so there are two equations. One is for substrate A, the guy we like. Okay, so what we're doing is calculating what's the rate at which this complex is growing, all right? Well, the answer is it's growing at 0 rate because we're at steady state. So however many bound complexes there are, that doesn't change with time to speak of. So it's getting fed by the on rate. And of course, there are some number of enzymes present. So each one is being fed, so we have such a term. And then it's losing stuff from the off rate and making the product. And of course, there are two kinds of things going on. We've got the good guy and the bad guy. So this is steady state. We let it run to steady state. And then basically, the only thing you care about, so you can solve each of these equations for, let's do it for one of them. We can solve for, okay, what is the concentration of the good complex? Well, I have an equation here. The steady state equation says the concentration of the good complex is just, I just rearrange the equation. All right? And I'm assuming, for simplicity, I'm saying the reaction rate doesn't really matter on whether I've charged the good thing or the bad thing. And that actually seems to be appropriate for all of the cases where this is biologically interesting. Okay, and we have the same thing for A goes to B. Now what do we care about? Well, we care about the error rate. We want to compare the rate of making the bad product compared to the rate of making the good product. So this is the error rate. So there's a similar thing for B. Just substitute B for A. And what's the error rate? Well, the error rate, okay, let's call it, I don't know, for some reason I started to call it F with a subscript 0. So I have to take the ratio, sorry, what is this? This is the reaction rate right? The good stuff and the bad stuff are both coming from the bound complex, and they're being made at a rate which is proportional to how many of the complexes in question I'm interested in. So the error, the fractional error rate, the ratio of the good rate to of the bad rate to the good rate is just this ratio. So really it's just the ratio of the bad complex to the good complex, and we have an expression for that, right? And so to say, it's the denominator thing that varies from one to the other. The numerator part, by assumption, is essentially the same. There's a common concentration of the enzyme here, whatever it is, and you know, we're going to say that the concentration in the soup of the A or the B is the same, and the on rates just have to do with diffusion, and they're not particularly different. So this numerator factor is going to be the same, both for the good thing that I'm trying to add or the bad thing, okay? So the numerator is the same, they'll cancel out, and basically all this can be, or what it is in the end, is just K minus A plus V divided by K minus B plus V, right? This is the expression you saw yesterday. And, okay, this velocity is a positive number. This is necessarily bigger than Ka, off, you know, the off rate for A and the off rate for B. And when we studied what these off rates actually depend upon, they depend upon the binding energy of the substrate to the enzyme. And all you have to do, all that you need is to have minus EB minus EA divided by KT. All right. So basically, there are other factors, but they're presumably of order 1. The only place where you can get a really big number is from this Boltzmann factor having to do with the energy difference. And the energy difference is, if you think about it, to look at the cases that we're interested in, it's a hydrogen bond or two. It's at most 10 kT, right? So a covalent bond is a whole electron volt. That's 40 kT for room temperature. So these energy differences are large compared to the thermal energy, but not that large, right? It's like e to the minus 10 twiddles e to the minus 10 twiddles 10 to the minus 4, and in many, many circumstances, that's just not enough, okay? That's just too big of an error rate. So the algebra is actually not that complicated, all right? And I don't know if I made that clear last time. So that's the problem. Something like this is going on when you're copying. There is, you know, we had some questions after the class about whether there could be interference from other pathways, and the answer is maybe. I'm not so sure. But the idea is, for everything that you want to, with great discrimination, add to some growing polymer, there's a specific enzyme that's supposed to do that. And our problem is, how does that specific enzyme that's going to pick out A, right, how can he be confused by B? And in the end, it's just this energy difference that matters. Okay, so that's the setup of the problem. And what else to say? Okay, so now the Hopfield-Ninio suggestion is, well, let's do it twice. Check once, and then recheck, right? Do it a second time, and hoping that basically what you will get is the square of this discrimination factor. And if you could do that, then you're getting down to accuracy numbers which are comparable to what you actually see in at least replication of the DNA, for example. And that is the most critical place. All right, so let's go the Hopfield-Ninio scheme. John, I was an assistant professor. No, I wasn't. I was a postdoc or something like that at Princeton, where John Hopfield was a professor, and we were kind of friendly. And I had lunch with him one day, and he said, "You know, I think I've figured out something really interesting." And he explained this whole thing to me, and I have to admit that, okay, it sounded good, but it didn't really make a deep impression on me. I can't say that I did not understand a word he said. No, I understand the individual words, but it didn't really sink in. And so eventually, a very large number of years later, it has finally sunk in. I finally got what he was talking about. And it was a very good idea. So and it turns out that this French theoretical physicist, Jacques Nignot, had done the same thing more or less at the same time, and they were completely independent. It's just a good idea that percolated up in the mind of humanity in independent places, right? This happens a lot, actually. Okay, so good idea whose time had come, and you know, looking back at, there's an interesting historical question. Why the hell did it take so long to sort this out? People really should have been aware of this problem, and maybe were. I haven't really dug into, was anybody else explicitly aware that there's a problem here of fidelity? Okay. Okay, so the next scheme. So I have enzyme plus substrate. So I'm going to do the same things. Enzyme plus substrate, and so I bind it. And then I come in, so this can go both ways. And then the idea was, well, let's suppose I use up some energy to do something irreversible, so that I mean, what is the substrate? The substrate is just a bigger molecular entity than the enzyme itself, and you know that you can modify the shape, energy, and whatnot of proteins, big molecular entities, by attaching a phosphate group, for example, to it. Then you change things, and its function can change. So I just put a star, and there's some rate associated with that. I'll just call it n plus. And then this guy, the second time round, can either make the product or not, as the case may be. So he has a K, so the second K minus here, I just want to, so here the backward rate is K minus S, and I'll just give it a different name, L minus S. So there are two places where there's an off rate of either the good or the bad, and it seems sort of obvious that this is going to work, right? Because the discrimination will come in making the off rate for the bad guy a lot bigger than the off rate for the good guy, right? That's what made everything work over there. And clearly here we're doing it twice, so I ought to get a better result. Okay, so one wants to check that. So the kinetic equations are a little bit more complicated, but they're not that much more complicated. And this basic observation that all that matters really is the ratio of the concentrations of the bad to the good bound complex just before it gets to make the product. All right, so what we're going to be, well, okay, well, we'll see it. Okay, so we let it run to steady state, and there are two places where steady state is established. The concentration of the, the original bound complex, that's now constant. I keep feeding stuff in. I mean, we're assuming that the concentrations of these substrates that can get added to whatever I'm working on are big, so that the fact that you've eaten one or two of them doesn't change the actual concentration, so these on rates don't change. Okay, that seems reasonable. There's a plethora of these molecules. Eating a few of them does not change the statistics at all. All right, so I have to tell you that there's an equation for this guy being in steady state, so to say a constant number of these guys, and ditto for this. So I have two equations. That is, I have two equations for each possible choice of S. Here I had one equation because I only had one bound complex that I was worrying about. So there was really only one steady state equation, but I had to write it for both the good substrate and the bad substrate. Here, because there are now two bound enzyme, substrate bound to enzyme states, there are two different states that I'm worrying about. I'll get two steady state equations for each of these possible substrates. And we'll see how that works out. Okay, so for a given substrate, I'm going to write, so first I'm just going to write, let's suppose S is the good guy, which I've been calling A for some reason. So I have two equations, one that says this guy is in steady state, so stuff is going on the enzyme, and stuff is going off. It's going off in two ways. It's going off by falling off with this off rate, K minus A times EA. But it's also going off because this irreversible step can happen, right? So that's another way you can lose these guys. You can lose him by M plus times EA. And there's a similar equation for this excited complex. 0 is equal to M plus EA. Okay, so this guy is being fed from this one at some rate. I don't know what it is, but we'll see what the conditions are. And then he's losing stuff. He's losing stuff to falling off because he's rejected. And making the product. Okay, so what is this going to do for us? Well, I have two things that I'm trying to figure out. Namely, in the end, what I'm really trying to figure out is what is the concentration of EA star? Because that's where the product is being made, and I'll need to compare how much of the starred bound complex there is for the bad guy as compared to the good guy, and that the ratio of those is going to give me the error rate. All right, so I have to do a slightly more complicated calculation. So here, this first equation allows me to write what's EA. And so this says that the concentration of this first fellow is, and so this is just straightforward algebra, right, is K plus AE divided by K minus A plus M plus, and EA star, the second equation, is according to this, m plus EA divided by L minus A V. So I'm just rewriting the steady state equations. One of them tells me that the first bound complex concentration is something that involves numbers that I know that are fixed. And the second one, the second bound complex, which is the one that's critical because it's from him that the product gets made, is some factors times what's the concentration of the first bound complex. So I just have to chain these two together to get that EA star. Okay, so this is several steps, but the algebra is really quite transparent, and I think you can follow it as I'm writing in the blackboard. So this is going to be m plus over LA minus plus V. That's this factor times EA, which I've already written, so that's K plus concentration A, concentration E, divided by K minus A plus V. All right? And now, all I have to do is to take the ratio of this guy to this guy, right? Sorry, B. So if I want to know what's the concentration of the starred complex, I can, to get the concentration of the starred complex, I just take all of this and replace A by B. And for the same reason as before, the error rate is basically just the ratio of the bad guy bound in the second form to the good guy, right? Just the ratio of this equation to this equation. So this is just the same guy with A replaced by B above. And we can see the things that are common and are going to divide out are everything in the numerator, right? Because it's the same irreversible rate for both of them. For simplicity, we're saying that these on rates don't matter. They're the same for both. And we're saying for simplicity that the concentration of the molecules that we're trying to add to the growing chain or be selective about, those concentrations are the same. And of course, well, it's the same concentration of the enzyme, whatever it is, all right? This concentration of the enzyme is not the same as the total enzyme concentration because some of the enzymes are being used up in these bound complexes. But it doesn't matter. It's just some number, and it's the same for both the A and the B. So I therefore get to write, finally, not so hard. So the error rate, let's call it F1, right? So it's V E B star divided by V E A star. This is the rate that I'm making the bad stuff. This is the rate that I'm making the good stuff. And the ratio is what I care about. And the only thing that has happened then is that where I had previously, it was this factor involving the off rate from the first complex that was the thing that determined everything. But this guy now comes with a similar factor coming from the off rate from the second complex. So this ratio finally is going to look very simple. It is going to be K minus A plus V, and he always comes with a factor arising from the second state of the bound complex, and divided by K minus B plus V, L minus B plus V. Okay? All right, so we did exactly what we hoped would happen. That is to say, by chaining these two together, we got an error rate that is the product of factors like this. And this is this is the discrimination you get out of one of these events. And I just have two events, so I get two discrimination factors. And here it is. And of course, with a little bit of fiddling, we show that what if this ratio is bigger than k minus a over k minus b. In other words, in order to get the lowest possible value of this, you have to take the smallest possible value of v. You have to take v small. The rate at which this reaction goes has got to be small. And in fact, it has to be smaller than the slowest of any of these rates. Okay? Anyway, so that's the ultimate. That's the best I can do. L minus a over L minus b. And each of these factors involves an off rate from some bound complex. And for each of them, the argument that what matters is the exponential of the energy, free energy difference divided by kT. That's true. Okay, so this time you got two factors. You got a factor from the first enzyme and a factor from the second enzyme, or from the second state of the enzyme. We, of course, don't know in detail. Presumably, these ratios won't be exactly the same, but the rough order of magnitude suggests that they will be something like this. Okay? And that's now we're golden, because this means that this second order scheme gives me an error rate which is basically the square of what you got from the first order scheme. So that's pretty cool. Okay? And so the numbers are such that, okay, if you're a living organism, you could live with that. You have, well, if you're a bacteria, you can live with that because you have a few million bases in your genome, and if you have an error rate of 10 to the minus 8, this is an error rate per copying a base in the genome. And the numbers that seem to be appropriate are of the order of 10 to the minus 8. So in copying a whole bacterial genome, maybe you have a probability of somewhere between a few percent and 10% of making one mistake. All right, and the bacterium can live with that. All right, Darwin can live with that because this is a natural mechanism for making small changes in the genetic state of a presumably successful animal, and making small changes. If they're bad, if you've really done something stupid with this one mutation, then that particular descendant of the original bacterium is not going to survive. And if you made a mistake that actually makes something good possible, changes for something that's more fit, then, well, maybe that bacterium will sweep the population and, so to say, change what the species actually is, all right? So, but you have to be careful not to be changing your genome too fast, all right? Now, viruses are different. Viruses actually have error rates that are as if they didn't do this kind of second-order proofreading at all. And so when a virus is multiplying, depends. When viruses are multiplying inside your body, copying mistakes are frequent, and that seems to be an essential part of the viral lifestyle. Okay? And it's kind of interesting because an HIV for the AIDS virus is actually particularly mutable, and inside an AIDS patient, you have viruses, but the viruses that are present inside one victim of this AIDS virus actually are kind of a cloud in sequence space. They're very, how shall I put it? There's a great deal of variation even internally. So, and this gets into the subject of evolution, which maybe we will get into shortly. Okay, now, let me just one last thing here, or a last thread. We made a very strong assumption here, namely that this is a one-way process, all right? Now, you could say, all right, suppose that just said, suppose I imagined that this process can even go backwards. So the original thought was, let's make it irreversible. We know that that's going to cost us energy. Okay, this is an important process the cell can perhaps afford to expend energy. So just for completeness, what would happen if you allowed this transition to have the possibility of going backwards and forwards? And what would happen if you let yourself imagine that you could do this in an equilibrium fashion? Would it work? In other words, is it possible this all will work if the transition from bound complex 1 to bound complex 2 is something that just can happen spontaneously? Why not? All these other transformations are happening spontaneously. Okay, and then, well, you would have to have a forward and backward process here. This would be, in fact, we could even do it here. So this is K plus. And it could be possible that this goes backward too, just like here. That is to say, you could go from free enzyme and substrate directly to this guy. That's a possibility. So if you wanted to look at the most general thing, right, you would look at something like this. So we decorate the two-step process with all the possible ways that the steps that can occur are reversible. They can go one way or they can go the other way. And we can then look at what would happen if we insisted that this was an equilibrium process. Then we could ask, what is it that I have to do? If I wanted to make it non-equilibrium, to drive it towards the irreversible process that gives us the result we want, I have to modify this in some way. Now, the way it's normally thought that what's going on is that this process, the forward versus backward, is driven by coupling it with some ATP consuming process, so that the forward and backward rates don't have to be equal, and the M plus and the M minus can be very different, and you can drive this pair of steps by coupling in some energy consuming mechanism. All right, but suppose we leave that as something we look at once we've looked at the whole process, what is the most general process going on. Okay, now here you have a much more complex set of reactions, but the logic is exactly the same. There are a bunch of parameters. What you ultimately want to do is to calculate what's the ratio of this, the concentration of this second complex for the good guy as compared to the bad guy. And you'll write down the equations that tell you in steady state the total rate of change of this guy has to be 0, and the total rate of change of that guy has to be 0. So there will be, exactly as before, for each of the substrates, there will be an equation that says that the rate of change of this, the number of these guys present is 0, and ditto for him, and it'll involve all of these, it'll involve all of the constants, and then the concentrations, and so on and so on. Okay, so it's a, this time, complicated, no, I mean, it's a slightly complicated algebraic problem. You have to, in each of these equations, in the equation that involves this fella, the concentration of this fellow is present, and likewise. So it's, I want to get the concentrations of the two states of the bound complex. And the equations that I write down two equations, but each equation involves both of these guys. So I have one of these nasty things where I have two equations and two unknowns. So I have to do some, the way you solve it, right, is to say, okay, I have two unknowns, x and y, and there's some equation that I have for, let's call it capital X and capital Y. These are the things that I'm looking for. These are the things that I know. And, you know, and then there's some matrix of coefficients that's acting on the two things that I'm trying to find. So what you have to do is to work through the solution of this. What I need to know is these two guys. This is ES, and this is ES star, let's say. All right? These are the things you want to know. And I have two equations that express steady stateness, and so but these guys are mixed together. There are two such equations, but each equation involves both of the unknowns. Okay, so this is just a complicated, nasty expression. This is a very standard look for a problem like this. The X and the Y are things involving the concentrations and the on rates and so on. So I just have to solve it, right? And the way you solve it, you know this, is that, okay, the thing you want, ES and ES star, right? If this is a matrix, it's just the inverse of that matrix times the stuff you know. All right, this is the algebraic procedure. If I have a matrix A, B, C, D, the inverse of that matrix is easy to write down because it's only 2 by 2. It's you exchange these guys, minus B minus C, and 1 over AD minus BC. This is the determinant of the matrix. Right, so even though the setup is more complicated, you can solve for the things that you need to know in order to get the ratio of the bad rate to the good rate. All right, and it's a little bit hairy, but not so bad. And so in the notes, it's worked out. There's no point in, I think, trying to do it on the blackboard. Could be done, but I think it would not be instructive. Okay, so let me give you the answer. All right, so what you're looking for in the end, and I'll write out the answer just to say it looks like an unholy mess. And trying to understand what it's saying is something that's hard to do analytically. All right, so anyway, the thing that governs the error rate, as we just as before, right, is the ratio of the concentrations of this second bound complex. And, all right, so we can write it out, and I'll just do it for old time's sake, and we'll leave it and we'll think about it. So just to give you a flavor of what you're looking at. L plus B, K minus B plus M plus times L minus A. Okay, I'm writing this out for a reason, even though it's probably not terribly intelligible. M plus M minus K minus A. That's the numerator, and the denominator is exactly the same thing with A, whoops. Well, sorry, let's just write it out. Yeah, it's basically the same thing with B replaced by A and A replaced by B. So this is M plus K plus A, L plus A, K minus A plus M plus, L minus B plus B, K minus B plus Okay, so when we got the end result, so this is now our error rate. Previously, when we got our end result, it was in a form that was pretty interpretable. That is, we could see what's going on. Here, with this more, a generalization of the second order scheme, the end result as a function of the parameters just looks like a mess, right? And how you're able to think it through, how you can manage to extract meaning from it is not clear. In Hopfield's paper, he says, okay, if you insist on thermal equilibrium, all of these guys are in detailed balance. For each of these steps, the forward rate and the backward rate is the same. That's what equilibrium means. He claims that even though you've got two steps, this actually, the smallest it can be was our first order rate. You have not done anything. Even though you've chained together two of these enzymatic steps, if you insist on thermal equilibrium, then you don't get any gain. Putting two together, two is not better than one. That's the claim. That's somehow buried in this complicated expression that depends in a complex way on the parameters. All right, so this is where, let's see, I guess there's one more thing I need to say, but it's kind of amusing to look at a simulation of this whole story that with all of these parameters in, just to see what happens and sort of extract some intuitive meaning from all of this. Okay, one more thing has to be added, and that is that all of these, there are lots of parameters. They're not entirely arbitrary in the sense that you must, if you have thermal equilibrium, there must be something called detailed balance. And I just want to give you a quick idea of what that entails, and then we'll have a look at the simulation. All right. So, as I say, the parameters are not completely arbitrary. What is it that I'm detailed balance? Okay, so detailed balance is just a statement that the rate at which you go this way from the bound complex to the excited bound complex is the same as the rate at which you go back, right? All right, that's different from saying you're in steady state, right? It's the statement that each of these possible transitions has the property that the net is zero right here, okay? It's not, okay, so what does that mean? That means that m plus times ES divided by m minus ES star, it's got to be 1. In other words, so the concentration of this times the transformation rate m plus, that's got to be equal to the reverse reaction, the concentration of the excited guy with the backwards transition rate. That ratio must be 1. Similarly, you've got two other such things. You have L minus S, ES star, divided by L plus S times S times E. And then that's got to be 1. And similarly, K minus S times ES divided by K plus S, E times S. That's got to be equal to 1. This looks, so what is this telling me? It involves these concentrations of the enzymes and the bound enzyme complex. Well, you can get rid of these things, which aren't directly just parameters that you're putting into the system, by doing various ratios of these guys. You can divide out the ES over ES star, and you can divide out the product of S times E, and so on and so on. So you can reduce all of this to a condition on the, so ES star over ES, just rearranging these guys, is M plus over M minus. And there is one thing that's left over, and that's K minus S over L minus S times L plus S over K plus S. So this is an actual condition on the parameters. This is what the detailed balance requires. And of course, there are two possible choices for S. It can be the good substrate or the bad substrate. It can be A or B. So actually, what I'm going to do is write it out for both of them. A, and K minus B, L plus B over L minus B, K plus B. So, you in order to be correctly constructed, the parameters that you put in, the kinetic parameters, have to satisfy this constraint. If it's in thermal equilibrium, all of these things must be the same. Now, if you mess with thermal equilibrium and make that ratio different than these, you can do that. And that's taking the system out of thermal equilibrium, and you can indeed physically do it by coupling this step to something that involves hydrolysis of ATP. Okay? So, all right, so I say all of this because I'll now show you something that you can play with yourself if you're so inclined. We can do a simulation of this whole process, and it's worth, I think, looking at it. So there are two things to say. So let's simulate the process. What do you get? So let's see. Let me, okay, now I want to share the screen. Okay, so can I do that? Okay.
Just click on here.
I think I did it.
Share, then you open.
Okay, why is nothing happening there? Okay, good. Okay. All right, I think we're good. Now I have to find my way through. Is it this? Yes. Okay, good. All right. Let's go up.
And then you click in this.
Yeah, right, okay. Okay. So on the Google Drive, there's there's a paper by Phil Nelson and a couple of collaborators in which they developed a very nice simulation of this process. And I just want to give you a quick, first of all, in order to actually do a simulation, you have to pick some numbers for all of these parameters. And so they constructed a set of parameters that are reasonable, biologically reasonable, that satisfy, to the extent that we want to, the detailed balance condition. And what does the simulation actually do? So you follow the enzyme, and it can be in one of 5 states. It can be sitting there unoccupied. It can have bound substrate A. It can have bound substrate A and be excited, or it can have bound substrate B and be excited, or it can have bound substrate B and then be excited. So there's essentially 5 different states in which the enzyme can be. And the simulation basically involves, this is a particular, a simulation of a particular realization. What are we doing here? I think we're building a protein, and we're incorporating, we hope, the right amino acid. So what the simulation amounts to is, given all of these parameters, having carefully chosen them so that they're semi-realistic and instructive, not in some insane far off corner of parameter space, you follow what happens to the enzyme, right? So if the enzyme is just sitting there, like so, what can happen to it? Well, it can bind one of the substrates, okay? It can bind either A or B, and you even have a rate at which it binds. You have put in a number, so you actually got a rate. Okay, so there are two things that can happen. It can bind substrate A or it can bind substrate B, and there is a rate for each. Okay, so what does it mean by doing a simulation? All right, well, you know that there's only two outcomes possible. So the next thing that will happen is that the enzyme will have bound one or the other of these two molecules. And you have the total rate for this to happen is the sum of the rate to bind A and the rate to bind B. Okay, so instead of actually advancing the time microscopic step by microscopic step, you say the time that has to elapse before you bind something is exponentially distributed. All right? You have a rate at which something is happening. The total rate for something to happen to the enzyme is the sum of the two different on rates. And so you say, I will draw from the appropriate exponential distribution a time. And that's the time, this is a statistically correct time for the event to happen. The event being, I bound one of the substrates. And then you say, okay, the event happened, but which of the two possible events? Well, I have the relative probabilities of the two things, and so I flip a coin. In this case, it's 50-50. So I flip a coin and I decide it was an A or a B, depending on the coin flip. So that's another draw from a probability distribution. Okay, now I have, I have one or the other bound. So I've changed the state of the enzyme from enzyme empty to enzyme bound to A, let's say, is where I am. Now, what can happen next? Well, it can either go to the excited version of the bound guy, or it can go back. So there are two events. So the next event is going to be one of those two. Each of those possibilities has a rate, and therefore the total rate at which something happens, the next possible thing, is the sum of the rate in that direction and the rate in that direction. So I draw from the appropriate exponential distribution the time at which the next event happens. I decide which event it was by saying it's 3 times more probable that it goes forward than backward, given the numbers that I've put in. So I flip the appropriate. I pick a number between 0 and 1 at random, and if it's bigger than the fraction that is associated with going, how shall I put it? An event happens and, it is 3 times more likely to go that way than that way, according to my numbers. So having said an event happens, I now decide which it is, this guy or this guy, by drawing a number between 0 and 1. And if it's greater than a quarter, I go that way, the one that's most probable. If it's less than a quarter, I go the other way. So, and then you just chain this together. This is what's called the Gillespie algorithm, okay? And it's very cool. And so a simulation then is simply, whoops, did I lose it? No. Okay, what happened? If I move, I cause it to...
No, I changed it.
Oh, you changed it, okay, all right. So, in this case, I've got this set of 5 possible states of the enzyme. And the simulation consists of going through these steps that I just described. Okay, you have to write a little Python program in order to, or whatever. In the old days, you would write a Fortran program, but these days you do something much more pleasant to, in a random fashion, so to say, modulated by these probabilities, you wander around between these states, right? And so a simulation is simply a sequence of such things, all right? And one of the possible outcomes eventually, of course, is I incorporate either A or B into my growing polymer, all right? And so how long it takes to get the next polymer, the next element incorporated into my polymer is something that's going to be, the time involved will be something that wanders around stochastically also. But anyway, the simulation is simply follow the system as it wanders from state to state, and you say, I want to make a polymer that's 1,000 units long, or 100 units long, or 25 units long. I just run until I finally added the 25th polymer, and then I look, and I ask, well, how many of the elements in this polymer are A? If they're all A, then I'm really very happy. Are some of them B? You look, and of course, depending on the numbers that you've put in, different fractions of them will turn out to be B. Okay, so let's see if we can actually see what happens when we do this, all right? And so what this program does, okay, so let's go back, let's go, I want to escape, yep, okay, so I think we don't need that anymore. All right, so now, all right, so what I'm going to do is to run I don't do this very often, that is to say, run a program in real time. So, notebook. Whoops. Okay, let's see, what have we got? Jupyter. The lights are dim and I can't type correctly. Okay. Therefore, what am I looking for? Star I, Pi, and B. Whoops, here we go. All right. All right. So this is the program that's adapted from this paper by Nelson and friends. And I'm running it in a Jupyter notebook. But is your Python skill up to running? I mean, so Python is a computer language, and you can, you'll write a program .py for Python, and then you execute it. Okay, great. And you wait and you see what comes out, if anything comes out. But you can do it in a notebook style in which it's broken up into cells, each of which has a collection of statements, and you can walk your way through it. It's much more convenient for developing and actually sort of quote unquote doing science. You can hop around, vary things at will. It's much more flexible than the straightforward running of a Python script, but you can do that. When you get into production mode, of course, you want to have something that you can send off to the central computing server and let it do its thing peacefully without you watching. Okay, so I think, what have we got? So the first thing basically loads libraries of some use. If we're going to do a plot, you have to have the matplotlib, blah, blah, blah. Mostly NumPy. Okay, so there is the heart of the thing is a function which basically does the Gillespie algorithm. There's a set of statements in which you initialize, and maybe we can go there. Initialize. Now, okay, let's see. Okay, so he sets things up in such a way that you have different options for initializing. You can set it up as a hop field ninio thing, what the situation that we talked about at the very beginning, where basically the transition from bound enzyme to excited bound enzyme is essentially, it's out of equilibrium. It's close to irreversible, but we have the ability to change parameters. Okay, so anyway, I've selected the simulation type as hop field ninio. You can do an equilibrium simulation in which this process does not get any help from the outside, and it is chosen to be in equilibrium, blah, blah, blah. So I probably won't go into that. Okay, so let's see. And the heart of the matter basically is that there's a transition matrix that you have defined by your choices of parameters. Namely, you know, there are these 4 states, 0, 1, 2, 3, 4. There are 5 states, excuse me. There's the empty enzyme, there's the enzyme with A bound, there's the enzyme with A bound excited, et cetera. So, and the parameters say that for any state from 0 to 4, there's some probability in the next step of making a transition. There's a rate for making a transition to any one of the other 5 states. Many of them are 0, right? Because if you're here, if you're in this guy, well, all you can do is to go to the excited bound enzyme or back to the unbound enzyme. So many of these transition rate elements are 0. Okay, so what I've added here to what Nelson et al. Have is a couple of little helpful steps which basically take this horrible formula, okay, and evaluate it. So this is the Hopfield prediction, if you will, for what the error rate will be in terms of all of these parameters that you had to put in. The thing that's really helpful about this paper, besides doing it and giving us a nice bit of code to modify, is that in a rational way, we try to identify values for these parameters that are quasi-realistic. And you have to go through the literature and figure out what's likely to be big compared to what. And that's a very useful and, how shall I, it's not in the moment all that rewarding thing to do, but they did it for us. So we have a set of numbers which makes sense, all right? Okay, so I just coded in. Let's calculate what this thing actually should be with this choice of parameters, and then we can compare it with what we see, all right? So back to this. All right, this is really, okay. So at the bottom, I've actually executed this, this cell, and what do you see? It's very, not the easiest thing to read, but what these 2 functions that I added to the initialization are giving me. First, these 2 numbers are the one on the left, 0.044, blah, blah, blah. That's the Hopfield prediction, straight up, okay? And the second number is what the prediction would be if you just use this as a 1-step process, okay? That is just chopped off all of the rest of it, okay? And so what do you see? You see that the 1-step process with these parameters, just using only these, you know, the probability of error per step, this is the error per step, is big. It's 0.2. And the Hopfield prediction is pretty close to the square of that, right, 0.04. All right, so this is the prediction on the basis of the analysis that we've done of the kinetic equations, all right? And just for the heck of it, the next line is printing out the actual values of these ratios of parameters that are diagnostic of am I in equilibrium or not, all right? If I'm in equilibrium, all 3 of them must be equal. And if I've gone out of equilibrium by changing the ratio of the forward to backward excite the enzyme complex rate, well, it'll be different. So this is just some kind of visual manifestation of how far out of equilibrium you are. So the 2 things that have to be true still, the parameter ratios of these numbers right here, this 10, which is maybe 50 times larger, that's how far out of equilibrium with these parameters the system is actually, how close to irreversible this guy really is. All right, so this is the Hopfield-Lineal notion, and we're taking the full set of equations and letting it run. And okay, so what should we see? All right, so you run the Gillespie simulation. Okay, so this was all set up, predictions and so on. Then you go ahead and run the simulation. So what is it? It's set up to do the following thing. just start with everything at rest. The enzyme is empty, and now you do this series of random events according to the probabilities that are built into the system, and you stop when you've incorporated, in this case, 25 units, okay? And you do that 10 times just so that you can get some statistics. Now, if you run it, it'll take a while, and in fact, for some reason it's set up to print out what transition you're doing at each step. You know, at each step, you're in a state and you're making a transition to another state. And okay, you roll the appropriate dice and you get the answer. I was in state 3 and I chose to go to state 2. And for some reason we're not keeping track of the time, but we could do that. Okay, when we're all done, we have the complete history. We know where this guy went from time, you know, from tick to tick. Every time there's a transition, I record where I went, where I started from and where I went, and I could also have recorded how much time that took. And what I'm doing here is just to look at the, I'm looking at some features of the output, all right? So this is one particular run in which I start from no polymer and I finish when I've got 25 units in the polymer. You have to look at the details. Well, what is it that I'm seeing? Okay, so the first two numbers are telling me how many times did a transition take place in which I actually added an element to the growing polymer chain, okay? And so this array is actually, well, sorry. So there's a states array, which is just the sequence of states, okay? 0, 1, 4, minus, da, 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 da. A minus means that's a transition in which something was actually added to the polymer, all right? And a -4 is I added a bad guy, and a -3 is I added a good guy. And I ran this thing until I made a polymer chain of length 25. Okay, so you can see that in this particular situation, when I put together 25 units, one of them was bad and 24 were good. And okay, so there is an error, and the probabilities of the errors, if you remember, was roughly 1 in, let's see, 0.05, so it's sort of 1 in 20 or so. So this is consistent with the Hopfield prediction for this model. And it's sort of the interesting thing. The interesting thing is how many times did I visit state 3 and how many times did I visit state 4? Well, 3 is the state I have excited I had a bound enzyme complex with the good guy and I excited it. I'm in the excited state of bound enzyme complex with the good guy. And state 4 is the bad guy is bound and to the excited complex. So this is counting, many transitions took place. There's a lot of jumping back and forth, but okay, what the second line is doing is counting the number of times you were in the course of this simulation in the different possible states, okay? Being in state 3 means I'm almost ready to add a good element to the growing polymer chain. Being in state 4 means I'm almost ready to add a bad element, okay? Okay, now every time I am in either of those two states, I have used up an ATP, you know, however many ATP molecules are associated with this transition. I'm out of equilibrium and I'm driving this with energy. So it cost me 50 tries. That's what oh, sorry, and so these are the counts. How many times was I in those states? I was in the state where I'm ready to add a good guy 236 times, and I'm ready to add a bad guy 50 times, all right? So I actually added 24 good guys. So that's like per added good guy, it cost me, I had to be in that state ready to do it and have consumed some ATP 10 times per thing that I add. Whereas when I added a bad guy, I only added one, but I was 50 times in the state where I could have, and I had to expend energy to get there. So what this is all saying is that if you say, in order to get myself into the state where I'm ready to add an element to the growing polymer chain, I have to expend some energy. All right. Now, if you say, well, I added 25 elements to the polymer chain, so I had to, whatever this quantum of energy is, I had to do it 24 times. No, I had to do it many times. I had to expend that energy many, many more times than the number of elements that I added to the polymer. This is the kind of thing you can sort of begin to get your hands around if you look at a simulation like this. Now, you know, the error rate is very large because we wanted to have, in a finite time simulation, you know, short enough that you didn't get bored and go watch TV, waiting for it to finish, to see actually some errors occurring. So these are not realistic numbers. The realistic numbers are way, 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 way smaller, but they're also way, way, way bigger in the, sort of say, energy inefficiency. So this is a really, it's a powerful process for achieving fidelity, but it is very wasteful of energy. Okay? Now, if you're copying the genome, I think you don't care. But it's not just wasteful of energy, it takes time. In other words, having to go, you know, try to add something 250 times and actually adding 24 times. You know, you're slowing things down. And that of course, was implicit in these formulas. In order to get down to the lower limit for the error rate, you had to make the velocity of the reaction, the speed at which it actually produces the output, you had to make it close to 0, all right? So it turns out that this whole question of speed and fluctuations and all the rest of that continues to be controversial in the sense that, you know, when you apply this scheme to specific things other than just copying DNA, sometimes you're really interested in making this happen accurately and rapidly. And, you know, the question whether this, how shall I put it, this scheme actually is good enough to cover what we're seeing happening is still up in the air. Anyway, so that's kinetic proofreading. It's a very important, so to say, conceptual issue with many things in modern biology, and this is one conceptual path towards a solution of how do you get adequate fidelity, adequate accuracy. Okay, so I went into this in more detail than probably is appropriate for, or how shall I put it, consistent with transmitting understanding, but I think this is one of these things where you really have to look in detail at the argument in order to grasp what it actually is good for. Okay, so that's kinetic proofreading. I'm going to switch gears. So first of all, if people have any questions, I would recommend that you try running Mr. Nelson's program with my little amendments. How many of you, you know, in sort of say the course of a week will fire up Jupyter and run some Python code to address some simulation or other problem? Are you familiar with this? Yeah, okay, good. Oh, that's great. All right, the more the better. Phil Nelson also has he has a great textbook on biophysics, and he has a follow-up which is basically simulation in simulation methods in biophysics, in which he develops how shall I put it, the ideas and code to implement them for a wide range of problems in biology. So it's well worth looking at. I just bought it recently. I haven't really worked my way through it, but I will eventually. Yes? I can't hear. Now I will be able to.
Okay.
Is it on? Yeah, it looks like it's on.
I just wanted to ask you to write the name of the suggested reading that
okay.
you just told us.
All right, so the paper that I was referring to by Nelson is on the Google Doc, okay? Now you wanted the name of the textbooks? Okay, I think on one of, let's see, my mistake. All right, so in this PowerPoint, tell you what, I will add, so there's 3 or 4 books that I can recommend. Okay, immediately after the lecture, I'll go and add them to this PowerPoint, so it will be somewhere in the PowerPoint up towards the beginning on the Google Drive, okay? All right, that way I'll get the actual title, the author's names, and the date and whatnot correct. Okay, all right. Maybe, so I'm at a fork in the road about what I'm going to talk about next. So I think I'm going to change my mind and talk about, all right. Here we go. So I'm going to proceed. Is that up on the screen? Okay, that's good. And now the question is, how do I get you, where's the view slideshow? Good. All right. Okay. So the word evolution was mentioned, and I want to get into, I'd like to explain the ideas behind a paper that I wrote some time ago with my colleagues on a version of Darwinian evolution in the genome of bacteria and yeast, okay? And I've said that, you know, one of the things that happens when errors are made in the copying of the genome is that, okay, you create a new bacterium which is slightly, you know, it has a slightly different genome than the one it started with. There's been one mistake somewhere in its 10 bases, and maybe it's a completely irrelevant mistake, or maybe it's a mistake somewhere in the middle of a gene and causes a change in the amino acid, and maybe that's a terrible mistake, in which case this new mutated thing will die. But maybe, you know, just maybe, it's a change that makes this new bacterium fitter for the conditions that it has to live in, and in which case it won't die, and maybe, it will actually grow faster than its brothers and sisters and eventually achieve a state of what's called fixation. In other words, you have a little pool of water somewhere on the shoreline, and in that pool of water there's some large number of bacteria that are all from the same species, and they're living together, and the conditions are such that it can support a population of some size, and all of a sudden, so you guys do ecology, right? Many of you. So this notion must be something you talk about a fair amount. So a mutation occurs spontaneously, and so suppose that the mutation is such that the mutated bacterium, now just one individual in the population, most likely, has a slightly greater growth rate than the original guys. This can happen. And actually, we don't know all that much about the statistics of this. What's the likelihood? You know, how many times do I have to have a mutation event that creates a new genome that is slightly fitter, has a slightly larger growth rate than the original genome? Okay, but let's suppose it has happened and the new guy has a slight fitness advantage, fitness being identical with growth rate here. If the population were infinite, then this guy with the slight advantage would eventually, almost certainly, expand to dominate the population, fix. In a finite-sized population, fixation can happen or not, as the case may be, and often more rapidly because of there are statistical fluctuations, and these statistical fluctuations in the number of offspring can be exploited by the slightly more fit organism to actually cause it to eventually crowd out everybody else. So this mutation can fix, right? And there's a very interesting story which I will talk about tomorrow, whether I will actually, there's some mathematics behind it. The question is, if I have a population of some size, and I have one guy in the population who, by virtue of one of these random mutations, has a slight fitness advantage, what's the likelihood that this new guy will eventually be the whole population? Okay? What's the likelihood that he will fix? There's some mathematics behind that. And in this paper that I will talk about, we exploited the mathematical results about the likelihood of fixation if you have a population of a certain size and a small fitness advantage. What's the likelihood that this guy will actually become the new species, okay? All right, so that's, and there's a very interesting context in which we can discuss this issue. All right, so the context is gene regulation in cells in general. So what do I mean by gene regulation? Okay, so we know that the genome consists of the code for genes themselves, so code that can be translated into sequences of amino acids that can be assembled into proteins, all right? That's the, so to say, the most important information. But you know, even in E. coli, there's something like 5,000 genes. So 5,000 stretches of DNA that, if transcribed and then translated, make an actual protein that can do something useful. So the 5,000 places where this can happen. In us, there are like 20,000 genes. Okay, but surprisingly, you know, more, but not all that much more. Only a factor of 5 or 6 or something like that. You certainly do not want all of those genes to be expressed all the time. So, you know, in different conditions, you need different things present. I mean, for example, take bacteria. Bacteria get their energy and their, so to say, the elements from which they construct themselves in order to reproduce. They get it from, among other things, sugars. And there are a whole bunch of different sugars. So how does a bacterium eat? Well, we talked about that at the very beginning. For every sugar that it could eat, there is a surface receptor that is capable of binding molecules of that sugar and essentially internalizing them, putting them inside. And so each of those surface receptors whose utility is to eat this or that or the other sugar, well, it's a protein, and it's coded for somewhere in the genome. And there's no point in making all of them all the time. You only want to make whichever one is appropriate for the environment you're living in. So you have to be able to regulate the expression of genes. How does that happen? Well, so here's a cartoon. In addition to the genes themselves, the so-called coding regions, naturally these guys are not bumping, the genes are not, so to say, bumping up one against one another. There's some dead space between them. In bacteria, not so much, hundreds of base pairs maybe, but there's space where you're not actually coding for anything. But what you do in those interstitial spaces, basically, is to have special sequences of DNA whose function it is to attract and bind things called transcription factors. What are transcription factors? Well, they're proteins. And what does a transcription factor do? Well, when it binds to its conjugate sequence of DNA, typically very near the beginning of the coding region of a particular gene, it essentially enhances the rate at which the RNA polymerase that will read the gene and turn it into an RNA message. It basically turns on the gene. So there are proteins out there called transcription factors. In bacteria, there are hundreds of them, and each one, well, first of all, it has to be made itself, but we'll worry about that later. If it's there and some appropriate signal comes along, right, that says, okay, we are now in a region where there's a lot of glucose or mannose or whatever, fructose, something happens and that transcription factor gets phosphorylated, let's say, and now it's activated. And if it finds its conjugate stretch of DNA on the genome, it'll bind to it. And those are the so-called promoter regions. And they're always near the start reading, the beginning of the coding region. And they then turn on the transcription of that gene. And, you know, for example, it might be the gene for the surface receptor that can actually grab onto the sugar molecule and bring it into the cell. All right, so the transcription factor proteins bind to the promoter to help or hinder, as the case may be, the polymerase copying the gene to message. And, okay, so here energy comes in here because the idea is that the transcription factor has it's a protein, all right, but it has some well-defined region that is meant to bind to double-stranded DNA, and it's constructed in such a way that it binds preferentially to DNA sequences that have a certain characteristic. It's not quite, but we could think of it as a kind of lock and key. This guy will only bind if it sees, you know, 10 bases that are A, C, C, G, 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 T, C, C, all right? It could be completely specific. It's very difficult to make it completely specific. Typically, what's at issue here is the binding energy, because this is all taking place under the influence of thermodynamics and statistical mechanics. So what really matters is the binding energy of this particular transcription factor to, suppose it's sensitive to 20 bases at a time. In other words, it's it's linearly reading the genome, and it reads it, let's say, 20 bases at a time. And if it is stuck to the DNA and the 20 bases that it's sitting on are its magic sequence, then it's very strongly bound, and that's great. That means it will turn on, well, if it in fact is sitting on a promoter, it'll turn on the gene. Okay, now there's an immediate problem. So the transcription factor diffuses like anybody else, any other molecule. And it's going to find the genome, it's going to find the double-stranded DNA, and where it lands and where it, so to say, tests out what's its binding energy could be anywhere. And if it lands on a stretch of DNA that doesn't match the characteristics of that 20 base pairs that it binds too strongly, well, it'll come off. And it has to wander around in a stochastic fashion, just like we were talking about at the very beginning of the lectures, and until it eventually finds the magic sequence that it's designed to bind to strongly, in which case it can do its job. Okay, so there's an energy issue. So just to leave it for the end of the day, for each transcription factor, it has an energy function that is going to govern what it's really going to do. And what is this energy function? Well, it takes in a sequence of bases, let's say A, G, G, A, C, T, A, 1, 2, 3, 4, 5, 7, I don't know. Something like that. I give it the magic number of bases, right? It's found a piece of DNA, and that piece of DNA, it's found the appropriate length. It has some size and shape, and it'll sit down on a stretch of DNA that has a certain length. I give you the sequence, and I want to know what's the energy. You know, how many 3.35 EV or something like that. And so the transcription factor is characterized by a function that takes in a length of DNA and spits out a binding energy. All right? And so if you take this guy and run him over the entire genome, it's going to spit out different values. Right? And you can actually do a kind of histogram. If I just say, running this guy over the entire genome of DNA, of E. coli, what is the distribution of energies? And usually less positive energy is more strongly bound. And somewhere towards the bottom here are the sequences that we are interested in. They are the ones that give you a strong binding energy, okay? Okay, so we're going to talk about that. Okay, so binding energy, whoops, I looked the wrong way. Come on, all right. Okay, so when we pick this up tomorrow, we'll talk about how you model transcription factor binding energies, and mutations come in because if in any particular species, as time goes on, the sequences in these regions where the DNA binds, where the transcription factor binds to turn on the transcription, there can be changes in the sequence. And what can you, presumably the changes that either enhance or at least don't decrease the binding energy are going to be changes that are acceptable to the organism, and changes that cause the binding energy to get weaker are presumably going to be not acceptable to the organism. And the question is, can you follow that logic to find out something about the way evolution works in microorganisms, bacteria or yeast? And it turns out you can. It's kind of interesting. So we'll pick that up tomorrow, and it's going to be, once again, about specificity and binding energy. Okay? And we're going to throw in a little bit of evolution to make it interesting. Okay? All right. So see you tomorrow.